So for immunology, I've switched things around a little bit. So everything you're going to need should be in modules. So go into Canvas. Let me know, has anybody tried to get into Canvas and not been able to get in yet? No. Let me know if you can't. Email me. So I guess I should write that down, because if you can't get into Canvas, Jim.Thompson@sfcollege.edu, and so just email me. Okay. There's a getting started document that should kind of walk you through what you need to do, what book you need, things like that. Syllabus is also going to be an important document to kind of go through. Again, the syllabus is going to have when are we meeting. That might change, but maybe it won't. My name, Dr. Thompson. You can call me Jim, James, Dr. Jim Thompson, whatever. This is where I'm going to be as far as my office, room 102. This extension might change, so I will make an announcement and I'll change the syllabus if that does. But really, one of the easiest places to get a hold of me is going to be email. I'll try to keep on top of emails so that I respond to you quickly. But mostly I'm going to be answering emails during my office hours, which are right here. And so if you were to email me in the evening and you don't get a response right away, just kind of keep an eye out for a response during my office hours. Office hours, I'll be over there, so I'm going to be here full time. I'm teaching pathophysiology, anatomy, A&P2, molecular diagnostics, so I'm going to be here most of the time. Text, we're going to be using Kubi. It's a good text, but one of the major reasons we also use it is because they use it over at UF. So that makes it easier for us to transfer credit for taking immunology between here and UF by keeping it at least consistent with the text. So we're going to be using the Kubi Immunology 7th edition. You do need to have access to Launchpad. If you've taken AMP here, it's similar. It's a different publisher. It's Macmillan, not McGraw-Hill, but it's going to be a similar kind of approach to going in there. In fact, I'm hoping it works, but right here is a tab on the side where you can click on that and get into our course for the publisher. And this is, you're going to be taking, so I'll talk about that in a second. So that's really all you need is the text and then access online. You should be able to get access with a code from your book, I believe. So grades, because people like to get good grades and want to know what they have to do to get good grades. We have 5 unit exams, which are going to make up the bulk of your grade. So I've got study guides for them. They're in modules. So as you're going through, pull up those study guides, use them. I promise if you study from the study guide, you'll more than likely do pretty well. So the study guides are good to have when you're reading through the book, because you know, well, I don't see this on the study guide. Maybe I'm not going to spend so much time reading it, although if it's interesting, by all means. If it is in the study guide, focus on it. So again, 5 exams. I dropped the lowest. You had a bad day, you didn't do very well on one of the exams, I'm going to drop it. You'll notice we don't have a final, no departmental final, no cumulative final. Immunology is pretty, and we're covering the whole book. So it's pretty massive. Having a cumulative final, when I took immunology, I did not have a cumulative final, and so I'm not expecting you guys to do that. It's a bit much. So really focus on these chapters as we go through. Another 10% is going to be the learning curve assignments. Now those are the online ones that you're going to be doing. The nice thing about the learning curve, so it's kind of like it sounds, it kind of gives you questions. If you don't do too well, it throws more of those type of questions at you. If you do really well, it says, okay, they know this, and it moves on. So it may take a little longer or a little not as long, depending on if you studied ahead of time, if you read the chapter. So it depends how you like to study. Maybe you want to just jump into the learning curve, open the book, and do it that way. It's going to take you longer, and some people like to study ahead of time than take the learning curve. You're going to get 100% if you do the whole thing. So you're going to get 10%, 100% if you do the actual learning curves. So this will help you grade a lot. I dropped the 2 of these, so we do all of them. So you have to do all but 2 of them, or however you want to do it. Or you can do all of them. I suggest doing them all. The other 10% of your grade is going to be quizzes, maybe assignments, but mostly probably quizzes. These are going to be in Canvas. Okay, and I've got 2 of them up already for the first 2 chapters. I will try and throw them up as we go. So that's it for grades. Let's see. So really, make sure you get into Launchpad soon. Let me know if you have problems getting into Launchpad, because I want to get them dealt with as early as we can. I have had people take this class online. I don't recommend it, but so I take attendance. I'm going to take attendance today, and then probably next week, maybe the next week, and then after that I'm not going to be taking it every single day. So this is one of those upper level classes to where it's up to you to decide how important it is to be in lecture, and I'm not going to harp on you to be here for that. So that's syllabus. Go back over here. Now we've got our tentative schedule. So a lot of times, I'm not sure if I updated all the dates in our assignments. So if I did, that's great. Fine, go into assignments and look at the dates. But really, this is what I'm going to go by. And if I ever change anything in what we are doing, I will make an announcement on Canvas. So if any of these dates change, for example, Labor Day is not going to change, that's going to be on the 4th, but our first unit exam is going to be on the 11th. If that is going to change, I will make an announcement. But for the most part, you can see we're going to cover Chapter 1 today. On Wednesday, we're going to jump into Chapter 2, get through that. So you can see it's almost about a chapter a day. So I recommend reading through it. Get your study guides out as you're reading through them, and make sure that you're, if you come in having read the information, you're going to do better. You're going to understand better. Because I talk pretty fast, because you have a lot of information. So if I'm talking too fast, if you've already read the book, then it's going to click a little bit better. So here's the dates of what chapters we're covering, unit exams. And then you can see, and we're coming all the way up. And so I wrote final here, but that's just because we're going to do the last exam, because there's so much to cover. We're doing the last exam technically during finals week. Okay, but that's going to cover basically chapter, let's see, a lot of chapters. For the most part, I try to keep it minimal as to how many chapters we cover for each exam. This last one is a doozy, but we're really covering kind of more of the basics of each of these chapters. Okay. Any questions?
So lecture is not mandatory?
No, it's not mandatory. Although I say that, and then I'm going to get people I'm going to be here lecturing to the camera, and that's really not as interactive as I'd like it to be. So. Give you guys just a little bit of my background. I got my PhD in molecular and cellular biology, doing tumor immunology is what I did for research. I was doing translational research, trying to develop vaccines for patients, and one of the vaccines I helped develop went into the clinic, at least for studies. And then I went to do a postdoc and was doing tumor immunology, and then did that in the hospital, and then I also worked on autoimmune disease, type 1 diabetes, a couple others, and so I've worked on breast cancer, lung cancer, melanoma, things like that. So that's kind of my background, so this is why I enjoy teaching immunology, because it's kind of my field, and I really enjoy it, and I think it's it's exciting because, and as you'll see at the end of today's discussion or PowerPoint slides, we're still learning a lot about immunology. This is one of those fields where we don't know everything. And year by year, we're making new discoveries. There's new therapies coming out based on immunology. There are tumor vaccines coming out. There are autoimmune disease therapies based on the immune system. So it's really a field where you're going to see changes all the time. Technology is changing. Flow cytometry kind of has been one of the major tools in helping to understand the immune system, and that's been growing, and so we've got a lot of technologies that are kind of being born out of immunology as well. Antibodies are huge for all fields, and that kind of came out of immunology. And these things have happened within our lifetimes, if not within maybe the past 20 years, some of these things. So it's pretty exciting. So I enjoy it. So as you can see, if you go into modules, Unit 1, here's our lecture. So we can just open it up straight out of that. Download it. So this lecture we're going over, you guys can, I wouldn't recommend printing it out full size because that's a waste of paper and ink, but feel free to do whatever you want with these guys. If you want to study by them, I've seen people put like 4 of them into one page and print them out, so however you want to do it. That's much good. So what is immunity? Okay, it's just a state of protection against foreign pathogens or substances. Again, our body doesn't know what's a pathogen or what's a foreign substance. We just have a system that tries to attack anything that isn't ourself. Observations of immunity go back a long time, 2,000 years, maybe longer. But we have recorded history where people noticed that, for example, back during the good old days of the plague, there were people who survived the plague, were able to treat people who had the plague and not get it again. And they noticed this. So they noticed, oh, this person, he got the plague or she got the plague. They survived, and they're taking care of other family members who have the plague, but they're not getting it again. And they'll take care of somebody else who has the plague, and they're not getting it again. So we started to understand there's some type of immunity going on with those people. Can we generate immunity without inducing disease? So way back then, they were getting the plague, and it was hit or miss whether they survived. So that doesn't help out a lot. But we started learning about potentially vaccinating people because actually cultures outside of ours would do things like variolation, where they would take somebody who had smallpox, they would take the pus out from the wound, and then they would actually put that into a scar that they would make on somebody else. And yes, sometimes they got smallpox, but sometimes it gave them a weaker form of smallpox, and they would then be protected against it. So we started kind of thinking about this idea of vaccinating people. So way back, some Greeks coined the term immunity, but like I talked about. Also, 15th century, so the variolation I talked about was way back in the 15th century. Okay, so we learned about that. Edward Jenner in 1798, he was the guy who noticed that milkmaids who were less likely to get smallpox, and they would oftentimes get this lesser version of smallpox called cowpox. And so he developed a vaccine based on cowpox. So he would give us something that wasn't virulent, but then would, now, granted, Jenner gave somebody cowpox, they survived, great, it wasn't that bad, and then he actually gave them smallpox to see if they would die. So it wasn't very ethical, but we did learn a lot from it. Louis Pasteur, okay, he took it a little further, and he said, well, let's not go around giving, because I can't find something that's like cowpox for everything. Anthrax, the same anthrax that kills animals is killing people, so I can't really give somebody a different version of anthrax. But he found that if we attenuate it, and actually I think he did it by accident. He had some anthrax that he was growing. He had left it out, it overgrew. He forgot about it. He went back and he injected it, and it didn't do anything to, I think it was mice. And then that same mouse got injected with a healthy version of it, and it survived, whereas the other mice didn't. And so thus was born a vaccine that was attenuated, or something that it's a vaccine that it's a bacteria, but we killed the bacteria, or we treated it in some way so it's not going to divide. So he developed the anthrax vaccine, rabies vaccine, so a couple others, which were attenuated pathogens. So first human vaccination, 1885. So, this is, not that long ago. It's, not within our lifetime, but still pretty recent. Since then, we've developed vaccines for lots of things, and we have pre-vaccine. You can see, the cases per year of things like smallpox, and then after post-vaccine. So in 2010, 0. Let's see, didn't we have a smallpox case not that long ago? Yeah, so we'll talk about that a little bit as well. Diphtheria, gone down to 0. Measles, okay, we still have a few measles outbreak. We have some at Disneyland, didn't we, or something in California. Mumps, not as many, but still out there, but really reduced. Whooping cough, that's one we've had, I think even in the area we've had a couple outbreaks. Now whooping cough is a vaccine, and I was talking to somebody about that earlier, is some of the reason for that is we get whooping cough vaccines when we're young. We actually need a booster in our adult age, and very few adults have it. And has anybody here had a whooping cough booster? Oh yeah, somebody's smart over there. Okay, good, you get extra credit. No. I haven't either, but that's something you actually should get a booster in your adult age, because you probably don't have a lot of resistance. Now granted, we shouldn't need it because there shouldn't be people running around with whooping cough, but because our herd immunity, this is something that means we all are a herd, and if the majority of people get vaccinated, then we're usually pretty good. But if that slips a little bit, then there are going to be carriers here and there, and then people who, like us, who forgot to get our booster are going to get whooping cough if we come into contact with them. Pertussis, polio, we've eradicated it, and we're still trying to get rid of it everywhere. Rubella. So again, these are pretty good, pretty impressive results. As far as medicine goes, they're pretty miraculous. And most doctors would be really happy to see those type of numbers. So pretty good reduction since we've implemented vaccines. So if anybody wants to have a talk about anti-vaccines or why people are afraid to have vaccines, we can do that. But there's really not much to it, because there's not a lot out there that really says that vaccines are in fact as dangerous as people think. I think there's a lot of people who just see a correlation between, we started vaccinating and we're seeing an increase in autism, and because of that, maybe it's the vaccines that we're giving to people. But we also have better hygiene. That's a difference. So it could be a hygiene thing. It could be a lot of different things. So there's really nothing that shows that vaccines are going to increase autism. Most of the things that we're giving people in vaccines are fairly safe, innocuous, not even as bad as what you're coming into contact in the natural world around you. So most of us have come into contact with some pretty nasty bugs, and it hasn't caused autism. And so what we're trying to do with vaccines is very similar, if not identical, as we're basically giving you a bug. We're just making sure it's not going to kill you. Or we're giving you a piece of a bug or a piece of a toxin, which is the case in things like the tetanus, which really hurts. your tetanus vaccine, or your tetanus shot, whatever you want to call it, we're basically giving you, there's a toxin that is made by tetanus, and that toxin we take, we do one of two things. We either take a small piece of that toxin and give it to you, or we take the whole toxin and denature it and give it to you. That's kind of the older one, but the older one worked a lot better than the newer one. So, we can talk about that later too. So, you know, within just a couple, you know, within the past 100, 200 years, we have learned about the immune system. So, in 1883, Paul Ehrlich, Metchnikoff, they demonstrated cell-mediated immunity. So they actually, in the microscope, they were able to talk about these white blood cells engulfing things, these phagocytic cells. We also found that there's this non-cellular component. So we knew we had these cells that would go around and eat things, but we also knew that if we took serum or took blood and got rid of all the cells, there was some protective element within the non-cellular portion of that as well. And that we found out later was antibodies and complement, things like that. We'll talk about those later as well. And for a while there was a great debate. It was great back then. It's not a debate anymore. But some people said no, all immunity is cellular. And some people said all immunity is not cellular, humoral, we called it, because it was part of the humors or the fluids, and which is why we still today call it humoral immunity when we're talking about the antibodies or B cell responses, and cellular immunities when we're talking about things like T cells and NK cells, things like that. So there was this debate. It's one or the other, and it ended up being both. So everybody won. You know, they were both right. You need both, and it just depends. I mean, you can get by with one or the other, depending, but you're really better off with both. And then there were also things like, we noticed that there was protection, or our immune system tended to not want to attack us. And this was Ehrlich, Paul Ehrlich, he called this the horror autotaxicus, or that basically what that means is our immune cells are afraid of hurting ourselves. They're horrified of hurting ourselves, and so there's some type of recognition of self, which is true except for in the cases of autoimmune disease. And so we've been learning about that as well. So a portion of immunity involves both humoral or antibody responses and cellular components. Humoral immunity combats pathogens via antibodies. So a lot of you know what antibodies are, right? And they're produced by B cells. Antibodies can be transferred between individuals to provide passive immunity. So here's something that you can actually get passive immunity. Babies get passive immunity from mom. They get antibodies from mom's milk. And they're not making their own antibodies, but mom's got pretty good memory of what kind of pathogens are out there. She makes antibodies. The antibodies go into the milk, go into the baby, and help protect the baby. You can get passive immunity by getting injected with immunoglobulin from people that say, I'm going to the Congo. I could get, actually, they would take plasma from people in the Congo, they would give me their IG antibodies, and I'd be pretty well protected against most pathogens in the area that aren't native to here where I'm at in Gainesville. So I could get passive immunity. But if I went back to the Congo a year later, I wouldn't have that protection anymore. That's why we call it passive, because you're getting antibodies, but you're not getting the cells that produce those antibodies, and there's no memory, and we'll talk about that when we talk about B cells in the humoral response. Cell-mediated immunity, that involves primarily T lymphocytes, or cytotoxic T cells. These T cells go around, and they look for cells that are basically infected with viruses, and or very small bacteria, and they kill the cells. Yeah, it sounds a little bit defeatist, but we don't want cells to be these nice little incubators for viruses. We don't want cells to be nice little incubators for bacteria, so we're better off telling those cells, hey, you know, it's time for you to end your life and get rid of them, than to have those viruses and bacteria floating around and growing. So T cells go around, and if you think about it, it's really nice that we have both of those because, let's see, well, we'll talk about it in a second. We have a lot of different kinds of pathogens. Some pathogens antibodies work better on, and some pathogens T cells work better on. So it's good that we have both. For a long time, we knew there was something protective in the serum, which are antibodies, but we didn't know how they worked. And this guy has been really big in the immunology world, Paul Ehrlich, German scientist. He saw this, he kind of theorized, which is pretty smart. I mean, he figured it out before we ever actually could see these things. He kind of theorized that there's these molecules going around and they have this lock and key type of mechanism where they'll fit something. And we make a whole bunch of different ones and they have this lock and key mechanism and they go around, and if they fit onto something, they're going to get rid of it. And so it wasn't until 1972 that Porter and Edelman determined the antibody structure. So and proved that he was right. He was the reason how these antibodies work is basically it's like Legos, or I don't know, something other than like lock and key was a good example. That one antibody it physically fits onto a molecule, and that gives its selectivity. There was another theory that, more of an instructional theory, that we had these kind of globular protein, or globular because we didn't even know they were proteins, things floating around, and they would become educated. They would form to fit things, and that ended up not being a right theory. So don't spend too much time worrying about that. So here's Ehrlich's kind of idea that you had these little swimmers, or these little guys, these little look like green onions, right? And they come around and they would fit onto something, and if they recognize that particular thing, they would bind and basically take it out. And that is very, it's not 100% true, but it was really close to being the case. So this is just kind of all of us mammals, mice as well. In fact, mice have an immune system very similar to humans, which is why we use them for research. So we all come into contact with foreign proteins or infectious agents, and we can have either B cell responses or T cells. We usually have one or the other. Don't usually have both. Sometimes we can have both, but usually we tend to have one or the other. And these, for example, B cells, they will basically, we have an antigen or a bacteria here, and sometimes B cells will bind to them directly. Sometimes those will be, and when they recognize those bacteria, something on that bacteria, they'll start to divide and become what's called a plasma cell, and then that plasma cell will just start to spit out antibodies. And those antibodies will just circulate throughout the whole body, and that's your humoral response. They'll find any other bacteria that are around, they'll bind to them, and then they'll work in conjunction with a couple other systems to get rid of those bacteria. We also have T cell responses here. For example, T cells, we actually have two types of T cells. We have helper T cells and cytotoxic T cells, and we'll talk about those guys. You don't have to memorize anything about them right now, but just in general, you have T cells that they'll recognize things like peptides or proteins or some antigen, and they're going to respond, and they're going to secrete cytokines. Cytokines are these little, you guys know what hormones are. Similar to hormones, but they act locally. So the big difference between a cytokine and a hormone is a hormone acts kind of throughout the whole body, and a cytokine is very local in its effect. Because, for example, let's say you have inflammation, you don't want your whole body inflamed, you just want inflammation in that one site of infection to call cells there. So cytokines are secreted, and they interact with these B cells or these T cells. So here we have, even though these are cellular, these are cells, they interact with B cells and help them make antibodies. So a lot of times when we say there's two separate arms, they do work together in conjunction, which is why it's important that we have both. So these helper T cells will kind of, let me get my pointer, laser pointer, so me flailing my mouse around is a little more visible. So we've got these T cells, right? And they will recognize an antigen. They recognize it a little bit in a different way than these B cells, but notice also that these B cells, their antibodies are actually their receptors. So you guys have covered receptors in anatomy and physiology. So B cells, they're receptors that bind to something and signal inside the cell, are actually the antibodies. And then notice that they are now, instead of using those antibodies as receptors, they're just kicking out the receptors and the antibodies can do their work. T cells, they don't do that. They have a receptor, a T cell receptor, that will recognize the antigen. We'll talk about how it does that. It does it in a different way. It usually sees antigen that's presented on a different cell, a professional antigen presenting cell. So they usually see antigens that are being kind of shown to them, a little bit different than antibodies. And they'll have a response and they'll excite these cytotoxic T cells that will recognize cells that have these little antigens and are presenting them. So it's basically like your own cells are constantly just basically recycling what's in them and holding little chunks of what's made inside the cell, outside the cell. And then T cells come around and they probe and look at what chunks of things are you presenting on your cell, what are you wearing, you are what you eat kind of thing, and if you happen to be wearing lots of chunks of virus, let's get rid of that cell. And so that's your T cell response. So those are kind of the two arms of that. Pathogens. So it's important to understand kind of just major groups or categories of pathogens so that when we talk about humoral or cellular responses, you can think about, well, which one would I rather have, or which one would my body rather have? So like viruses, examples could be poliovirus, variola virus, we've talked about some of these, causing smallpox, polio, things like that. T cells tend to be more suited to getting rid of viruses because viruses don't usually hang out outside the cell a whole lot, and they're really small. But they do like to go into cells, and so T cells usually get rid of those. Fungi, we usually have different antibodies that are really effective at helping to get rid of different fungi. We got yeast, Candida albicans, we have other things. And for most of us, you're not too worried about things like yeast, but if you're immune suppressed, yeast could be very, very bad. So we're constantly getting rid of viruses and fungi. Parasites are large. Okay, so for example, comparing parasites and bacteria are pretty small. Bacteria are smaller than most of our own cells. So bacteria are usually, we can get rid of them with phagocytic cells that will engulf them. So antibodies usually help facilitate that. So bacteria are usually, B cells are great for getting rid of bacteria. Whereas parasites, we don't usually engulf parasites. So you have usually other cells that will kick out granules and make the environment really toxic to kill the parasite. So again, different types of pathogens are going to be treated in different ways. So that's why it's really important that we have this varied ways to respond to pathogens. So just pictures like examples. So you've got this parasite, it's very large, that would not be easily phagocytosed, whereas a virus usually is going to divide inside of a cell. You've got bacteria, very small, and here we see, I believe that's a phagocytic cell that's going to probably engulf those bacteria. And here we have small, but yeast budding. So again, just keep in mind that these are all going to be dealt with in different ways. Let's see how we do it. Okay, we got about a couple more minutes. So immune responses rely on recognition of molecules. So we have kind of two approaches to this. We have an old school approach that we kind of developed early on in evolution, and it's been around for a really long time, which is where we have these pattern recognition receptors that recognize, so we have what are called germline encoded, which means that we're all born, you, me, and we all have pretty much the same all the same pattern recognition receptors. We have neutrophils that have receptors that recognize certain things that have been around for a really long time, things that are expressed on bacteria, and these are called, they bind to things that are called these pathogen-associated molecular patterns, or PAMPs. So these two, they're in red because there are two things you need to kind of memorize, because we're going to talk about them all throughout the semester. So pattern recognition receptors, if you read through the book, a lot of those are what we call toll-like receptors. Don't worry about where that comes from. It comes from flies and something else. So, but these are basically pattern recognition receptors. We all have them. They bind to things that are common. Sugars that are only expressed in bacteria. Certain lipids that are only expressed in bacteria. So again, these are things that we don't make, but lots of bacteria make them, lots of pathogens make these things. So we have kind of what we call this innate immunity against pathogens, and that's pretty much our innate, what we call our innate immunity consists of these PRRs that recognize PAMPs. Yeah, question?
Do any of those target endonucleases, which are found in like E. coli specifically?
Yep. Some of them recognize double-stranded RNA that we don't have.
Oh.
Things like that are virus. So there are viruses that have. There are some that recognize nucleotide epitopes that we don't have. Certain methylated epitopes that are found more commonly in bacteria and so there's a lot of them, and they recognize some of these commonalities. But yeah, so some of them nucleotides, some of them lipids, some sugars. There are quite a few out there. So, and they recognize these generic molecules found in many different types of pathogens. Peptidoglycans are one. So these are going to be different than things like antibodies, which are more randomly generated. We're going to talk about antibodies, how when we make antibodies, the antibodies I make are probably different than the antibodies you got, than you have, and it's based on just randomly taking a bunch of these building blocks and just rearranging them in crazy different orders and just throwing them out there. And we throw out billions of different variations of these antibodies in hope that they recognize something. So it's a very different approach than these PRRs, which are very specific, as opposed to randomly generating B and T cell receptors. So both, when I say antibodies, we also randomly make T cell receptors, and these are part of what we call the adaptive immune system. So we have this innate immune system that it's innate because we're all born with it, and it's all the same. And it recognizes things that are pretty common. But bacteria are tricky little buggers, right? They grow really fast, they adapt, they mutate, and so we've adapted to respond to things like viruses and bacteria that have mutated to get around our PRRs that have gotten sneaky and can sneak past those. We've evolved to have this more adaptive way to recognize things that get by these PRRs. And that's why we call it an adaptive immune system. So we have both this innate, but we also have an adaptive immune system. Okay. So humoral and cell-mediated immunity relies on surface receptors, B and T cell receptors. Kind of talked about that a little bit. This lock and key mechanism where they recognize things that are on bacteria or on virus particles, things like that. These are randomly generated by DNA rearrangement in B and T cells. So we don't start out with them in life. We actually rearrange genes. And this is one of the things that is very recent. Let's see, I can't remember the year, but it's been in some of our lifetimes. If you've been around for a while, it happened in your lifetime, where we actually learn. We always thought that our genome was fixed. Okay, it doesn't. We have genes, and they don't change. They don't switch information. But we actually learned that B cells and T cells, they'll take this long, crazy gene, and they'll just start rearranging it. And that's within your chromosomes. That's not mRNA. That's the chromosomes are actually your basic genome is being rearranged in these cells that are becoming B and T cells at a certain stage. So one B cell genetically is going to look different than this B cell, is going to look different than this B cell, at least in that particular gene that codes for the receptor. Same with T cells. So it's pretty exciting that we figured out how do we have this incredible variety of antibodies. Because really, I mean, we couldn't have enough, because we're talking billions of different variations of antibodies. We couldn't, we would have to have a really big genome to have that many genes just sitting there. And it might be, wouldn't be valuable to basically use have all that genetic material just sitting around when the great majority of it isn't going to recognize anything. So, again, we make lots of different B and T cells. We call it kind of a shotgun approach, but most of them are non-viable or deleted during development. So either because they recognize ourselves, they don't recognize our certain things very well, they don't rearrange well, but and then sometimes they go out into our system and they don't ever find their match. They don't ever bind to anything, and eventually they atrophy and die. So, but if we do have a B cell that happens to recognize something that's not ourself, then it can start to respond to that thing. So it's a really interesting approach. And which brings me to clonal selection. So you've got B cells and T cells. You've got a lot of different flavors of B cells and T cells that have different receptors on them. And if one happens to find a virus or a bacteria or a pathogen or a toxin and it binds to it, it's going to become a clone, and it's going to expand, and it's going to make more of itself, and those are going to respond to that pathogen or that dangerous element. So this is due to each cell having many copies of a receptor on their surface that only bind to one type. So again, this is probably an important point here. Each cell has its own receptor, and it only has that one receptor. It has a lot of that receptor, but it's only one type of receptor. This cell doesn't have the same as this cell, They're all different, but they don't have two different types of receptors. So one B cell doesn't have two different antibodies. It has one antibody, okay? And that's important. So when a B cell or a T cell interacts with its specific antigen, so I've been throwing out the word antigen, and I never really defined it. All an antigen is something that is recognized by that receptor. If an antibody binds to it, it's an antigen. We're going to talk a little bit more go into more detail on antigens. But basically, it's just some foreign thing. It could be a molecule, it could be a protein, it could be DNA, it could be a lipid, it could be a sugar. Any of those could be antigens if they bind to an antibody or a T cell receptor. So once the B cell or T cell interacts with its antigen, it's selected and becomes activated. Activation results in now proliferation of that. So B cells and T cells are one of the only white blood cells that, well, not the only, but they'll go out and they'll divide out in the periphery if they find something. Things like neutrophils. Remember back in A&P we talked about white blood cells, and you have neutrophils, eosinophils, basophils, all those other phils, macrophages, things like that. They tend not to divide. You have what you have. If you need more, you make more. They don't divide out in the periphery, but B cells and T cells, they do when they come into contact with something. So they're a little different. So activation results in the proliferation, producing a large number of clones, and then those clones are going to react with more of that antigen and help to boost the immune response. But that's why when we talk about immune responses, adaptive immune responses, if you've never seen this thing before, it takes a while to make a lot of those cells. So that's why sometimes when you have a cold and it takes a whole week, and the doctors say, don't come in unless you've had it for a week, because we're probably not going to give you anything for it, because maybe it'll go away on its own if it's a virus. That's usually something you've never seen before, and you have to mount an immune response. You have to wait for those T cells to divide to attack it, get rid of it. Sometimes you get a cold and it only lasts for a day, 2 days. That's probably something you've seen before, and you have some memory cells that respond really quickly. Sometimes, you might get something that you get rid of pretty quick. Sometimes that might be thanks to the innate immune system. So things like the innate immune system respond very rapidly. If you get a cut, the innate immune system rapidly will respond. So we'll talk about that as well. But clonal selection, you have the stem cell. It makes a bunch of different, here we have B cells. So you have a stem cell, it makes this B cell that has, it's yellow, this pink guy, this blue guy. They all have different antibodies, different antibody receptors. This pink guy happens to find an antigen that basically he binds to, and by binding he sends a signal into the cell that says, hey, start proliferating. He starts dividing, and now I can have a lot of these guys that will go out and take out whatever that antigen happens to be. Okay, so, but it's also important that we have something called tolerance. This ensures that the immune system avoids destroying host tissue. It also actually, I know the slide says host tissue, but it also can protect against destroying good bacteria in your gut. So you probably all heard that we have a whole lot of bacteria in our body. And we have a whole lot of bacteria in our gut, and actually some of that bacteria does great things, helps to make vitamin D, things like that. So we actually need that bacteria, so we don't want the immune system to get rid of everything. So we have mechanisms of tolerance where, and this is also within maybe the last 30 years we've had people theorize, because we used to think that for some reason our immune system could tell self, non-self. We think that the immune system, some people theorize that we only attack something if there's a danger signal. Okay, so how do we know something's a pathogen? How do we know something's bad for you? How do we know something's a toxin? So there's usually, we believe that there has to be some type of danger signal in order to tip the balance into a response. And danger signals could be things like damage to tissue, it could be other cells that are alerting of danger. But unless we have that, sometimes we become tolerant to something. If you think about if anybody's had allergies and got allergy shots and got you're getting desensitized to that, whatever it is, that allergen. And we're trying to introduce that allergen with allergy shots without danger signals, so that we're driving more of a tolerance phenotype. So tolerance is really important to learn about, and we're going to talk about that quite a bit. Because the opposite to tolerance is we're going to attack our own cells. So there's mechanisms in place, lots of checks and balances to make sure that our immune cells don't attack our own cells, and we're going to talk about those. So there's things called central tolerance and peripheral tolerance. So there's lots of different stages at which we induce tolerance to ourselves, but maybe even to things like bacteria as well that aren't doing harm. So we use these two, and I've talked about these as we kind of went through. We have our innate immunity and we have an adaptive immunity. So the big difference between the innate and adaptive are one, the innate immunity, and when we say innate, we mean things like neutrophils and cells that have those PRRs on them that are going to respond very quickly. We don't have to wait for them to recognize things because we've got a whole lot of them floating around, and they all have a receptor already that recognizes lots of different bacteria. So these are our innate responders, and because we already have a lot of them floating around, because remember, neutrophils are the #1 most abundant white blood cell, so they're going to respond in minutes to hours. If you cut yourself, neutrophils are there very rapidly. Those are usually the first responders. Specificity is limited. It's fixed. It's, if it recognizes it, great. If it doesn't recognize it, there's nothing I can do about that. Response to repeat infection, same each time. You don't have memory. Our innate immune system doesn't have memory. We don't select clones that have a specific receptor. They all have what they have. Major components of the innate. So I talk about cells, but I kind of skim back past the fact that barriers are part of the innate system. Let's just not let the bacteria in. Let's have things like skin, kind of. Let's make our skin acidic, so bacteria don't like to grow on our skin. We have these phagocytes like neutrophils, macrophage. We have these pattern recognition molecules. So that's innate. Whereas adaptive, response time's not as quick. It may take a few days. We've got to wait for the cells to ramp up. But we get this really highly diverse adaptive to improve. And even, we'll talk about B cells. B cells might recognize something at first. They can actually mutate to where they recognize it even stronger and have an improved response over time and an improved memory response. Each time you get the same thing, each response is going to be faster, quicker and quicker, because you have more and more of that particular type of T cell or B cell still hanging out and waiting if it ever sees that infection again. So that's things like vaccines help to induce that type of memory in us, so that if we ever see that bacteria or virus again, we're protected against it. And these are mediated by T and B cells. So these antigen-specific receptors and antibodies. Okay, so innate immune response, we're going to go into this in more detail. This is just a kind of skimming through this. Innate immune response is the first line of defense. Very fast as mentioned, but not specific. Uses germline encoded recognition molecules. So again, germline just means my stem cells code for genes that code for those receptors. They're not changing. They're always going to be the same. They're part of the germline. And these guys use phagocytic cells quite a bit. They don't only use phagocytic cells, but that's one of the major mechanisms by which they get rid of stuff. The adaptive immune response is humoral B cells and cell-mediated T cells. It's a little slower, again, 5 to 6 days or more, somewhere around there, depending on what you're infected with, how robust your immune system is. And these use randomly generated antigen receptors. We're going to talk about how this happens. But it's very specific to individual antigen molecules. So again, we need multiple vaccines, for example, because this vaccine, the measles vaccine, isn't going to protect against polio, so again, it's very specific. Innate and adaptive immunities actually work cooperatively, so they're not completely separate, one evolved after the other, but they communicate. So the innate response usually is our first response, and it will usually say, hey, I'm taking care of this, but it'd be nice to have a little bit of backup. They use cytokines, they excite the adaptive immune response and help to boost the adaptive response. And the guy who is kind of one of the most important intermediaries between the innate and adaptive immune response are helper T cells. And so T cells are part of the adaptive response, but they can also help with innate. So all of these guys it's gonna be intertwined. Signal molecules stimulate and direct adaptive immune responses as well. So kind of a back and forth a little bit. All right, memory. Memory is a hallmark of the adaptive response. So what's the big? So again, questions that you could have on exams are, what's the difference between innate and adaptive immunity? So time, how quick, memory, big difference between the two, things like that. So really, when you study these, whenever I have two contrasting things, make sure you can kind of compare and contrast them. So memory is a hallmark of adaptive immunity. The primary response is initiated upon first exposure to an antigen. So this is what we call primary response. After that primary response, a lot of these lymphocytes that divided will hang around, become memory cells, and are left behind. They'll sit in the tissues maybe, or they'll sit in the bone marrow, and they'll basically wait until you need them. So a second exposure to the same antigen is going to re-stimulate memory lymphocytes, but we have, instead of starting with one, you're starting with tens of thousands. of cells. So it's going to be much more rapid to ramp that response up. And reactivation yields faster and more significant, better responses, especially in B cells. Again, B cells, the first time they respond, the second time they respond, their antibodies are usually have higher specificity, so it's actually a more robust response. Memory is not present in innate immunity. So here, this line, we're looking at the innate response. If you had this, notice how it picks up really rapidly, magnitude of the immune response. Picks up really rapidly in first day. And it's gone. And then if you have another infection, same bacteria, you have the same type of response. It responds really rapidly, but it's not that robust. Whereas adaptive may take longer for that initial primary response, but it's a little bit more robust. And then when you have a memory response, it's very, very robust and can respond almost as rapidly as an innate response. Okay, so let's see how many more slides I have. So here's an example of kind of an immune event that can happen. So number one, the pathogen is picked up by the phagocytic cells in yellow. And then those phagocytic cells undergo changes and carry the piece of the bacteria to a local lymph node. So you guys learned about lymph nodes and immune responses. That phagocytic cell that we're talking about could be a Langerhans cell in the skin. So remember the skin and the, stratum, basale, the stratum granulosa, maybe somewhere in between there, you've got sitting one of these Langerhans cells. And it picks up some bacteria that snuck past it. And it will then, once it picks up an antigen or a bacteria or something, there's danger signals. It tells the cell to start migrating to find the nearest lymph node, goes to that lymph node. The phagocytic cell releases cytokines, and they're going to cause fluid influx and help recruit. So you're going to get local inflammation. Your lymph nodes are going to start maybe getting sore. Helps recruit other immune cells to the site, so you get an influx of T cells and B cells. And then T cells and B cells get activated and start expanding. So this in the lymph node is where we're going to have that interaction. And when we talk about these type of responses, you're going to find that the reason for lymphatics, the reason for these lymph nodes, the reason for this place for T cells and B cells to hang out is because, again, we have a lot of different B cells and T cells, and rolling the dice and hoping that the right T cell meets the right antigen, and expecting that to happen just anywhere in the body, we're going to improve those odds. Okay, so we're gonna basically these T cells and B cells are going to be looking for things in the lymph nodes. Okay, so we're helping to localize things. And remember, the lymphatics are constantly flushing things through them and into those lymph nodes. We've got our cardiovascular system. About 10% of the fluid that goes out of our capillaries doesn't come back into our capillaries. It goes through the lymphatics, and it just flushes everything into those lymph nodes. And so really this is where our T cells and B cells are looking for the antigens that they're going to match up with. And then if they find them, they'll start to divide and then go out to the periphery and respond. So that's kind of the gist of but there are downsides to having a system that works this well. There are dysfunctions of immunity that fall in maybe two broad categories. There's overactive or misdirected immune responses. If anybody's had allergies, you know that that's really annoying. Asthma. And sadly, most of these allergies and asthma are against things that aren't really dangerous to us. So it's really annoying. Autoimmune disease, it's not super common, but it does happen. Multiple sclerosis, Crohn's disease, irritable bowel disease, certain other things. There are a lot of autoimmune diseases where our immune system mistakenly attacks our own cells. Type 1 diabetes is another. the other category is immunodeficiency, is the other side. Is sometimes we have genetic defects that affect our immune system, and it doesn't respond. Or a secondary or acquired loss of immune function due to things like infections, or coming into contact with things that affect your immune system, toxins, things like that, radiation, can affect your immune system. And that, so things like autoimmune diseases, things like immunodeficiencies can lead to an increased risk for cancers, things like that. So you kind of have, it's not perfect. The other problem that we didn't really evolve to have transplants, but we've learned through technology and medicine to how to give people hearts, things like that. Unfortunately, our body doesn't like to have things in it that are not it, and so we've developed this mechanism to get rid of anything that's not self, but yet we have to suppress the immune system in cases. And it's really hard to make something that is not seen as foreign, but actually they're making strides in things like pigs to make maybe pig hearts more human-like, and trying to put in things that will actually help us to not recognize them as foreign. So there's a lot of research being done to try and improve transplants, maybe targeting the immune system so that it affects rejection, whereas we're trying to not touch the part of the immune system that doesn't reject organs, things like that. So we're learning a lot about transplants, but it is one of those big problems with having an immune system is you cannot easily transplant tissue. And then cancer is another problem, situations where dangerous cells that we want to get rid of happen to be our own cells, and we've developed this mechanism of not killing our own cells, of our immune system not doing that. But the truth is that when our cells mutate to the point where they don't look like our own cells, our immune system is constantly getting rid of tumor cells. So our immune system is really good at getting rid of defective cells. It's just some cells will evolve to evade being recognized, and they use mechanisms that our own cells normally use to try to prevent immune responses. And so that's another big problem that we'll talk about. So, in summary, we got through the whole thing, which is surprising. Immunity is a complex subject. It's broken down into, we'll break it down into layers and focus on each area to make it a little bit easier. But it's also exciting because if we understand how the immune system works, anybody can contribute to the field and help to exploit this understanding to prevent infections through vaccination, exploit it to treat illnesses, shutting down immune diseases, ramping up anti-cancer responses, or provide safer organ and tissue transplants. And again, there's a lot of research in the works right now. And just to hammer home that this is one of those fields where you can still learn things. A lot of our big discoveries started in 1901, and we're still having big discoveries even as recent as 2011, and even more recently. I could probably add to this list. So, if you want to go through and look at it, I'm not going to talk about each one of these, but feel free to go through and look at some of the really cool things that we have discovered in the last few years. So that is it. I'll let you guys go home. And I appreciate you sitting through the lecture, and I'll try and keep it lively and exciting. Try and relate it to the clinic as much as possible. Some of the chapters it's a little hard, but we'll try to do it.